Today we are going to see about principle and process of biotechnology. Okay, so now biotechnology is the science of applied biological process. In other words, it is science of development and utilization of a biological process, forms and systems for the benefit of mankind and other life forms. The term biotechnology was coined by Karl Erecki, a Hungarian engineer in 1919. Biotechnology has been extended to include any process in which organism, tissue, cells, organelles are isolated molecules such as enzymes are used to convert biological or other raw materials to product of greater values. Development of biotechnology. Biotechnology has developed by leaps and bounds during the past century. The development of biotechnology can be well understood under two main heads, namely conventional or traditional biotechnology and other one is modern biotechnology. Now we are going to see about conventional method. It is otherwise called as traditional biotechnology. This is the kitchen technology developed by our ancestors. It is as old as human civilization. This technology uses bacteria and other microbes in the daily usages for preparation of dairy products like curd, ghee, cheese, and in preparation of a food like idli, dosa, naan, bread, and pizza. This conventional biotechnology also extends to preparation of alcoholic beverages like beer, wine, etc. With the advancement of science and technologies during 18th century, these kitchen technology gained scientific validation. Next, we are going to see about modern biotechnology. There are two main features of this technology that differentiate it from the conventional technology. They are ability to change the genetic material for getting new products with specific requirement through recombinant DNA technology. Second, ownership of newly developed technology and its social impact. Today, biotechnology is a billion-dollar business around the world. Pharmaceutical companies, breweries, agro industries, and other biotechnology based industries apply biotechnology tools for their products improvement. Modern biotechnology embraces all methods of genetic modification by recombinant DNA and cell fusion technology. The major focus of biotechnology are fermentation, biomass, enzymes, biofuels, microbial inoculants, plants and animal cell culture, recombinant DNA technology, process engineering. Now we are going to see about fermentation. Fermentation for production of acids, enzymes, alcohols, antibiotic and fine chemicals, vitamins and toxins. Biomass for bulk production of single cell protein, alcohol and biofuel. Enzymes as biosensors in processing industries. Biofuels for production of hydrogen, alcohol and methane. Microbial inoculants as a biofertilizer and nitrogen fixers. Plant and animal cell culture for production of secondary metabolites, monoclonal antibodies. Recombinant DNA technology for production of fine chemicals, enzymes, vaccines, growth, hormones, antibiotics, and interferons. Process engineering. Tools of biotechnology is used to effluent treatment for water recycling. Traditional biotechnology. As described earlier, it is the kitchen technology developed by our ancestors that was using the fermenting bacteria. Thus, it includes the process that is based on the natural capabilities of organism. The word fermentation is derived from the Latin verb fervere, which means to boil. Fermentation refers to the metabolic process in which organic molecules, normally glucose, are converted into acids, gas, or alcohol in the absence of oxygen or any electron transport chain. The study of fermentation, it is practically uses, is called zymology and originated in 1856 when French chemist Louis Pasteur demonstrated that fermentation was caused by yeast. Fermentation was caused by yeast. Fermentation occurs in certain types of bacteria and fungi that require an oxygen-free environment to live. The process of fermentation are valuable to the food and beverage industries with the conversion of sugar into ethanol to produce alcoholic beverages. The release of CO2 by yeast used in the leavening of bread and with the production of organic acids to preserve and flavor vegetables and dairy products. Bioreactor or fermenter. Bioreactor is a vessel or a container that is designed in such a way that it can provide an optimum environment in which microorganisms or their enzymes interact with the substrate to produce the required product. In this bioreactor, aeration, that is ventilation, agitation, that is mixing up very well, temperature and pH are controlled. Fermentation involves two processes. One is upstream process and another one is downstream process. Upstream process. All the process before starting of the fermenter, such as sterilization of the fermenter. Sterilization means to remove the bacteria or fungus from the vessels. Preparation and sterilization of culture, medium and growth of suitable inoculum are called upstream process. Downstream process. All the process after the fermentation process is known as downstream process. This process includes distillation, centrifuging, filtration and solvent extraction. Mostly, this process involves the purification of desired products. Procedure for fermentation. Depending upon the type of product, bioreactor is selected. A suitable substrate in a liquid media is added to a specific temperature, pH, and then diluted. The organisms, it might be a microbe or animal or a plant cell or organelle or enzyme is added to it. Then it is incubated at a specific temperature for the specific time. The incubation may either be aerobic or anaerobic. Withdrawal of a product using downstream processing method. Now, we are going to see application of fermentation in industries. Fermentation has industrial applications such as microbial biomass, microbial metabolites, microbial enzymes and bioconservation. So first we will see about microbial biomass production. Microbial cells like alga, bacteria, yeast, fungi are grown, dried and used as a source of a complete protein called single cell protein, which serve as human food or animal feed. The next one is microbial metabolites. Microbes produce compounds that are very useful to man and animals. These compounds are called metabolites and can be grouped into two categories. One is primary metabolites and another one is secondary metabolites. Primary metabolites. Metabolites produced for the maintenance of life process of microbes are known as primary metabolites. Example, ethanol, citric citric acid, lactic acid, acetic acid, etc. Secondary metabolites. Secondary metabolites are those which are not required for the vital life process of microbes but have value added nature. This includes antibiotics, example amphotericin B, which is taken from Streptomyces nodosas, penicillin, and it is taken from Penicillium chrysogenum. Streptomycin, it is taken from Streptomyces griseus. Tetracycline, and it is taken from Streptomycin aerophaciens. Alkaloids, toxic pigments, vitamins, etc. are all taken from the secondary metabolites. Microbial enzymes. When microbes are cultured, they secrete some enzymes into the growth media. These enzymes are industrially used in detergents, food processing, brewing, and pharmaceuticals. Example, protease, amylase, isomerase, and lipase. The next one is bioconservation. Biotransformation, or modification of the substrate. The fermenting microbes has the capacity to produce valuable products. Example, conversion of ethanol to acetic acid, isopropanol to acetone, sorbitol to sorbose. This is used in the manufacture of vitamin C. Sterols to steroids.